Good afternoon. I'm Dr. Neil Druve, Health Program Specialist at the NIH Countermeasures Against Chemical Threats program. Welcome to this webinar presentation on cooperative agreement funding opportunities. I will be joined by Dr. Chardell Spriggs and Dr. David Jett, respectively, the Program Manager and Program Director of CounterACT. First, a couple of logistics. I'd like to remind you that we are recording this webinar. I'm also going to share the slides with everyone. And finally, we will be taking questions through the Q&A box. Please enter any questions you have there. You can enter them at any time. After the presentation, Dr. Spriggs and Dr. Jett will answer as many of these as time allows. With that, we can begin the presentation. First, we will present you with an overview of the CounterACT program. We will then speak about the specific cooperative agreement funding opportunities. And lastly, we will share some tips and lessons learned for when you construct your applications. And now I'll turn it over to Dr. Jett.
Well, hello, everyone. Thank you for joining us on this webinar. The NIH Counter ACT program has been supported since 2006. And its mission is to understand fundamental mechanisms of toxicity for chemical threats, and then use that knowledge to develop safe and effective therapeutics to improve our nation's medical response capabilities. And again, to do this, we support both basic fundamental research as well as translational research to the level of optimized lead compounds. So first, what exactly is a chemical threat? Well, the program is guided by a list of about 200 chemicals developed by the federal government, and applicants should contact myself or Dr. Spriggs to confirm that the chemical or chemicals they wish to study are on this list before developing an application. These are broad examples of chemicals of interest. They include traditional chemical warfare agents such as nerve gases, toxic industrial chemicals, toxic agricultural chemicals such as insecticides and rodenticides, pharmaceutical-based agents, and other chemicals. Next slide. So we cannot at this time share the list of chemical threats with you, but we can discuss the list in detail with you to facilitate your choice or confirm your choice. One thing that is helpful that's not on the FOAs is our toxidromes. This is where we have grouped chemicals on the list based on their mode of action and primary effect. This not only gives you an idea of the kinds of chemical threats, but some medical countermeasures developed for one chemical may also be effective for another chemical threat in that particular category or toxidrome. These toxidromes include anticoagulants, blood agents, cholinergic warfare agents, cholinergic pesticides, convulsants, hemolytic, opioids, lower and upper pulmonary agents, and vesicants. So you might imagine this needs to be a truly trans-NIH effort. Because of the diversity of chemicals and their effects, several other NIH institutes and centers participate in the program based on their expertise needed to cover things like ocular, skin, pulmonary, and other effects of the chemicals we study. These other institutes include NINDS, National Institute of Neurological Disorders and Stroke, National Institute of Arthritis and Musculoskeletal and Skin Diseases, National Institute of Environmental Health Sciences, National Institute on Drug Abuse, National Eye Institute, and NIAID, or National Institute of Allergy and Infectious Diseases, that provides oversight and manages the contracts for us. Each IC has a program officer assigned that can help you with specific scientific questions.
The CounterACT funding opportunity announcements, or FOAs, are designed to feed into one another and promote a progressive translational program structure, beginning with exploratory R21 grants. Our lead identification FOA covers activities from confirmation of molecular targets to preliminary in vivo efficacy and toxicity studies. The goal here is to have at least one lead compound ready for optimization at the end of the project. If you have already identified a lead compound, then you should apply to the lead optimization FOA, where you will conduct the needed preclinical activities to prepare you for transitioning to non-NIH funding and advanced development, such as with an industry partner or the Biomedical Advanced Research and Development Authority, or BARDA, both of which are beyond the scope of the CounterACT program. Our U54 Research Centers of Excellence is the most involved FOA. It spans the entire spectrum of research that we fund and has multiple working parts, including synergistic research projects, scientific cores, and the newly added Emerging Science and Scientist program. In general, with each step in this progressive ladder, you get a little more support and a longer duration of funding. For the purposes of this webinar, we will focus on the Cooperative Agreement FOAs. Please make sure to visit our website at the address shown and click the CounterACT program funding opportunity announcements to make sure you are using the most recent FOA. Cooperative agreements are a NIH mechanism frequently used for high priority research areas that require substantial involvement from NIH program staff. CounterACT cooperative agreement FOAs are managed with milestones. Milestone-driven research is used to ensure the research is focused on a well-defined goal and that the research activities achieve that goal with greatest efficiency. Milestones or goals describe precise study outcomes, not simply study completion, such as testing 100 compounds. Milestones should provide quantifiable measures of success. A brief rationale should be included for the milestone to justify the parameters tested, assays or models used, and the quantitative values for the success criteria. Milestones are set on an annual basis and should progress each year to meet the end goals of the FOA by the end of the project period. A bad or unacceptable milestone would state to complete tests on a drug, but a better milestone has criteria included to unambiguously quantify success. For example, the milestone or goal is to demonstrate efficacy in reducing lethality when drug X is administered at 20 minutes after cyanide challenge. The quantified criteria for success is reduction in lethality in mice by 50% when drug X is administered IM 20 minutes following an acute dose of hydrogen cyanide via inhalation. Efficacy is evaluated at 24 hours after exposure. For more information on milestones, please visit our website under information for applicants.
As Dr. Spriggs mentioned, there are 2 cooperative agreement funding opportunities through CounterACT that are intended for individual researchers. These are the U01 grant mechanisms that support research to either identify or optimize a lead compound. I'll elaborate on the first one. The first U01 that we have supports research on the identification of lead compounds that can act as medical countermeasures to chemical threats. By the end of these 3-year projects, you will be expected to have a compound or compounds for which you have demonstrated that they are biologically active and feasible to produce. You will be expected to have shown that their action is specific with high affinity and potency, and that they are selective to the target. Finally, you will need to show that the lead compounds are efficacious and safe. You will be expected to demonstrate these in both in vitro and in vivo models. The scope can include confirmatory mechanistic research, but there should be reasonable expectation that the compounds you are testing could work. If your research is at a more exploratory stage, that is fine, but you might be better suited to apply for one of our R21 funding opportunities that Dr. Spriggs mentioned. Supported research can also include screening for candidate lead compounds, demonstration of biological activity and target engagement, proof of concept in vivo efficacy, and preliminary compound characterization, such as ADME-tox or pharmacokinetics and pharmacodynamics. As far as the timeline goes, the next round of these cooperative agreements will have a receipt date this September. They will be reviewed by March 2022, with the first awards being distributed in July 2022. These lead identification grants fund up to $300,000 in direct costs per year for up to 3 years, and there are no renewals allowed. Next, Dr. Spriggs will elaborate on the lead optimization FOA.
The next U01 is for more advanced studies where a lead compound has already been identified. And the goal at the end of the 4 to 5 year project is an optimized lead compound ready for advanced development activities that lead to regulatory approval and licensure. In this case, it is recommended that you find experienced collaborators and seek expertise in drug development. The scope includes, but is not limited to, efficacy in human relevant animal models. This should be consistent with the product's final intended use in humans, PK studies with validated analytical methods. You may establish acceptable safety margins and confirm lack of drug interactions, and also develop stable synthesis and formulation. More specifically, we have included criteria that, if met, should deem your project eligible for consideration by BARDA or other advanced development entities. Your project at the end of the 5-year period should culminate with a lead compound with well understood ADME, efficacy data in an appropriate animal model, stable and scalable synthesis. Your preliminary regulatory strategy should be well developed, and you should have intellectual property rights and freedom to operate. The receipt dates are the same for the other U01 program. They are September of this year, with review in March of 2022 and awards made in July of 2022. The difference for this program is that the budget and project period is larger. Here you may request up to $500,000 in direct cost per year, and the project period may go up to 5 years. And there are also no renewals allowed in this program as well. For these applications, addressing each of these criteria, perhaps in a table form, is helpful. A fictional example is presented here where you can see each criteria has been addressed and feature the status, such as complete or proposed, and where to find the proposed studies in the application. This serves as an excellent reference point in often long and complex applications. Sometimes it's hard to know which CounterACT mechanism to apply to. If you don't know the molecular target or have a drug candidate or assay or animal model, then you should apply to the R21 program. The R21 is also appropriate if you have a drug that has been approved for another indication and would like to see if it is efficacious as a therapeutic against chemical threats. If you know a little bit more, such as the target and mechanism, or have several drug candidates for down selection, but need to conduct additional ADME tox studies, then the lead identification U01 would fit best. Now, if you do have a lead compound and it hasn't been optimized yet according to the criteria, then you should apply to the optimization U01.
Okay. So finally, we also support large multi-component research centers of excellence. These centers are designed to facilitate interdisciplinary collaborative research. The scope of the research spans from target and candidate identification and characterization all the way through to candidate optimization. Now, because of the higher resources that NIH invests, these centers can only propose research on certain chemicals, which include the cyanides, the organophosphorus nerve agents and pesticides, opioids, chlorine and phosgene, and sulfur mustard and lewisite. The timeline is the same as the other U01s, September 2021 receipt, March 22 review, July 22 award. The difference for this program is that the budget is much larger, but the project period is the same, and that's 5 years, 4 to 5 years, usually 5 years for centers. So the center structure. So the centers consist of one administrative core for center management and operations, including the pilot project program, ESSP3, 3 to 5 interrelated research and development projects, up to 3 scientific cores if needed and well justified, and 1 research education core. A figure showing the overall center structure and the synergy among the different components can be very helpful. The program has oversight, an internal group, a steering committee, and an external advisory committee. And I'll just say that the first thing to consider for these applications is whether you have strong preliminary data and a team of researchers with projects that fit together synergistically around a central theme. Another thing about the education core, and that is the budget can be no less than $50,000 direct costs and should focus on an overall goal of increasing the number of capabilities of medical countermeasures researchers, building on the strengths of the center investigators. So if this is a new application for you, at a minimum, you should shoot for the identification of a lead compound by the end of the project period. If you're renewing a center, you should aim to have an optimized lead compound ready for transition to advanced development at the end of the period. And you should also show progress has been made in the last funding period and address things like how significant was the contribution to the scientific knowledge of the field, have the objectives and milestones been accomplished, and do you in fact have one optimized lead candidate? And there's no renewals for this as well. So for a little more information on the pilot project program, we expect to support about 2 to 3 of these pilot projects per year. The project support is up to $75,000 per year in direct costs. And this is not to supplement ongoing research of an established investigator.
So there are some additional considerations when preparing an application that we would like to discuss. Some recent changes at NIH need special attention. Please read the specific notices listed here on enhancing and implementing rigor and transparency in NIH-funded research. Pay close attention to describing scientific premise, now rigor of the prior research, specifically the quality, including strengths and weaknesses of data you include in your application. Present a rigorous experimental design, including criteria such as proper statistical analysis and control of bias. Address sex as a biological variable. If you cannot use both sexes or you see no need to, have a good explanation why not. Too expensive is not an acceptable reason. Address authentication of key agents used in your research, including the chemical threat agent. Saying you purchased something from Sigma or other supplier is not sufficient. You will need to run an analysis to verify the identity and purity of key compounds. The emphasis here is on key agent and does not include minor reagents and buffers. Do not use the vertebrate animal section for experimental details. Reviewers are not required to consider this section for that purpose, and this may result in less enthusiastic review scores. Some important CounterACT specific things to consider here are how the therapeutic you are proposing will be used in humans. Having practicality in real-world civilian mass casualty situations regarding timing and route of administration is important. This is different than military applications. In the civilian space, we cannot predict when an incident will occur. This means the final product should have utility for emergencies involving acute chemical exposures where medical intervention is required immediately, either in the field or in hospital. For more advanced programs, such as the optimization FOA, pre-exposure studies, including those proposed to demonstrate proof of principle bioactivity, are not considered responsive. We encourage working with drugs that are already approved for other indications, since sometimes the regulatory pathway to approval is less difficult. And you may want to emphasize the relevance of repurposing in the innovation section, including quoting directly from the FOA. Special consideration will be given to research relevant to subpopulations with higher vulnerability, such as pediatrics and pregnant women. Include the structure of your lead compound in the research strategy section. This information is often helpful in determining feasibility. The regulatory strategies proposed should address essential elements described in the relevant FDA guidance for industry documents, and links for these can be found on our website. Please work closely with your institutional technology transfer office throughout the research process to develop your intellectual property strategy. You will minimally need freedom to operate. More advanced projects should have a plan to secure IP by the end of the project period to ensure a path forward to advanced development. And finally, address biohazards, containment and disposal of, as well as facilities needed for research on restricted chemicals, such as chemical warfare agents.
Okay, so here are some further considerations. Scientific feasibility is key. You need to establish this early in the application. Preliminary data and the literature should clearly support the hypothesis and proposed studies. You must include sufficient details so that reviewers can evaluate the approach and, importantly, the rigor of your study for example, blinding and randomization. You should show a clear pathway such that the work meets the goals of the project. If needed, be sure to address interdependence of aims and give alternative approaches to research strategies. You should make sure you choose the appropriate FOA for your level of research. And finally, don't just say the key investigators will communicate. Describe exactly how and how often they will communicate. And this is especially important for the center applications. So in summary, cooperative agreements involve substantial support from NIH staff. The NIH program, CounterACT program uses cooperative agreements for translational research that leads to optimized lead compounds. Milestones are used to manage the project. Individual research project FOAs include the lead identification and lead optimization programs. Be sure to contact us for more advice, especially to determine if the chemical threat is acceptable. And as a note, the due date for cooperative agreements is September 14th, 2021. So I'll stop now and turn that over to Neil.
Thank you. In addition to the cooperative agreement funding opportunities that we have been talking about, I would like to bring your attention to a couple of relevant IC-specific FOAs. One is to support research that promotes a basic understanding of chemical threats to skin. Its first receipt date is October 16th, 2021. And for further information, you can contact Dr. Hung Sang at NIAMS. Another is geared toward understanding chemical threat agent-induced pulmonary and ocular pathophysiological mechanisms. This FOA has a first receipt date of October 12th, 2021. And for further information, please contact either Dr. Srikanth Nadadur at NIEHS or Dr. Humam Araj at NEI. As mentioned earlier, CounterACT is a program that reaches across many institutes of NIH. Each of them and their respective program officers with contact emails are listed here. If you have specific questions about how your research might fit with any of the ICs, either within the context of the cooperative agreements or with the IC-specific funding opportunities, we encourage you to contact the relevant program officer directly. Once again, I will be sharing these slides with everyone, so you will have all these names and contacts, and also all the embedded links that have been in the slide presentation. That brings us to the end of the presentation materials, and I'll now turn to the questions. Please submit your questions through the Q&A box. I'll give you a couple minutes so that you can type away. And when they come in, I'll read them, and then Dr. Spriggs and Dr. Jett will answer them. Okay. So the questions are starting to come in. Please keep them coming. We'll start with this one. Do you support pure computational research, such as repurposing of FDA-approved drugs?
I can take that if you'd like. So historically, we've had some in silico work supported in the earlier funding programs that we've had. The key here is with these two particular FOAs, if you think you can go from in silico work to a lead compound in 3 years, then that might be feasible, yes. But if not, you might want to look at some of the earlier R21 programs that don't have that endpoint requirement.
Okay, so the only other question that we've received came in prior to the presentation, and that is that they were hoping to hear about the key differences between the two U01s, the lead identification and the lead optimization. And that is what a major part of the webinar was about. So I do hope that we have answered this question, and if you continue to have some questions about that, you can follow up with an email. You can send an email to me, and I'll direct it to the person who is best suited to answer.
I mean, I'd be happy to elaborate if you like.
Okay.
I mean, the key difference is the funding and the funding period, you know, they're different. One's for 3 years, one's for 5 years, one's for more money than the other. But the key to the initial lead identification is really you're starting from having an idea of what your lead compound would look like to a point where you actually have a lead compound as defined in the FOA. But that lead compound is nowhere near ready for advanced development, where you have to do a lot more things that are related to regulatory requirements, including things like, you know, synthesis and stability and all those other things that you absolutely need for pre-IND kinds of work. So it's just another phase, that next step you need to get this ready for truly advanced development. And when we say advanced development, we're talking about, you know, phase 1 clinical trials on up to a drug approval. So there's a much more sophisticated and advanced program that at the end of the day, we're finished. It's no longer an NIH kind of project.
Okay, thank you. So we have had some more questions come in, and you know, I encourage you to keep sending them. So the next question is, if you could please elaborate on the pilot project funding opportunity.
Chardell, or I can take it if you want. So, I mean, this is kind of new. We thought that these centers could be a good place to bring in new people to the field, and new people in the sense that they're early stage investigators, finished up their postdoc and so forth, as well as experienced investigators who find this area interesting and would like to engage. And so these are short, 2 years, short-term projects. 2-year pilot projects, 2-year short-term projects, so they can get some preliminary data that would enable them to then apply for some of the more formal CounterACT funding opportunities. So it could be, you know, a research investigator, science investigator, or even a postdoc that's, you know, coming off their postdoc that could apply for this. The only catch is it can't be part of what they're already doing. It has to be something new. And I mean, you can read some more details in the FOA about this. Let's see if there's anything else to mention here. No, that's about it. And please, give us a call if you want to talk more about it.
Okay. So the next question is about the lead identification FOA. Does it allow for animal model development?
So for the lead ID FOA, again, the lead identification and lead optimization FOAs are really designed for you to develop your compound, whether you're doing some down selection activities in the lead ID or optimization activities in the optimization. For me, I think the animal model development should maybe come in R21 or maybe some very early stages of the lead ID. I can read directly from the FOA where it says a development and utilization of animal models to demonstrate preliminary proof of principle efficacy data. Animal models should demonstrate an important clinical benefit and should simulate the ultimate clinical use of the candidate therapeutics in humans. And for that, I feel like the animal model should be in place somewhat. And Dave, you can, of course, elaborate on that.
No, that sounds right. It depends on your timeline. I mean, realistically, if you think you can develop a brand new animal model and get to a lead ID in 3 years, maybe it can work. But I would suggest that you look at our website and look at all of the studies, both current ones that are active, and I think if you go to the NIH reporter, you can look at previous ones. We've been doing this for a long time. We have developed many, many different kinds of animal models that you might want to look at and maybe even get some tips on how to develop it or even use those animal models.
Okay. So the next question is, can you elaborate on the choice of route of administration for the identification FOA?
Sure. So we're dealing with civilian mass casualty situations, and that usually involves an exposure event. You call first responders, where first responders have to come to the scene, and all of this takes time. And so if you are proposing something that is, the antidote is administered at the same time as the exposure event, then that's not amenable to a civilian mass casualty situation. And we're also talking about mass casualty situations. So it's not one person that you have to treat, but there may be very few people that have to treat hundreds of people. And with that in mind, we're thinking something like an IM auto-injector is an appropriate route of administration for in the field applications. You want something that you do not want to set IVs in the field. Now, after evacuation in the hospital, an IV might be appropriate. And that's just the case you need to make in your application. Again, a lot of this is phrasing and how you make your case in the application.
Okay, thank you. So the next question is, if the lead compound IP is held by an external company, does that preclude it?
So we don't have rules that preclude compounds, and we do have investigators that work with compounds where the patent or the IP is held by someone else, and they can usually give a license to that investigator to conduct their research. And so you do minimally need freedom to operate, which may mean that you don't have to hold the patents, but you do have to have that license. And so I think that's what's important there, is that we don't have hard rules that preclude it, but we would like you in your application to be able to lay out a regulatory strategy and path forward so we know the investment will not end at the end of the project.
Yep, I absolutely agree. I mean, the key here is freedom to operate. A lot of times some companies want to see the data before they start jumping on board. And so they'll give freedom to operate, a license to work with their compound and see where it goes. And we've had several cases where it went well and moved on to advanced, and even cases where we've got a drug approval. So yeah, and in those applications, especially the optimization one, you have to address it and definitely talk to your tech transfer people on how to address that and what's the plan, to be redundant. Yeah.
Okay. So still a couple of questions about how to figure out or parse the difference between lead ID and lead optimization. So it's if you have a compound that is known to work as a medical countermeasure, but no one has been able to effectively manufacture the compound, would that fall under discovery or optimization?
Oh, okay. It's a great question. And I think that if you have solid data with the compound, solid data that approaches, that it is doable to have this ready to go by the end of 4 or 5 years, then it probably could fit into the optimization. Now, if you only just have some very preliminary data, then it might have to go. Then it might have to go back. It might have to be in the lead ID. But I guess the key is going to be the criteria that we laid out in the FOAs that specifically define what a lead compound is. If you can make that case, then you've got the lead already. You can go for the optimization. If you can't make it, then you would need to be with the lead ID.
Okay, great. So the next question is, for the optimization, is it that the investigator should focus on a maximum of one drug or one drug class?
I think here you will have to have sufficient preliminary data that would have probably allowed for some down selection. So it's not that it's limited, but you do want to propose things that are feasible and that make sense. We don't have a max limit, and we do have, investigators who have a backup. And so again, you might not want to come in with just one compound without a backup, because one of the, things are going to be, well, what happens if this doesn't work? So, Dave, you can probably add more to that.
No, that's exactly right. And I hate to keep harping on this, but I mean, if you've got a class of compounds and they all sort of meet the lead compound criteria, then you can go into an optimization. But by the end of the study, you got to have your champions. You may have a backup, but you have to have your champion that you intend to go into advanced development with. Remember, at the end of that optimization, you have to have a target product profile. You have to have a lot of specific kinds of information. And so that's usually for one compound, not a class of compounds.
Okay. One more question about the differences between the lead or the different FOAs is one person asks if the reviewers are trained to identify the specifics of each FOA, and I guess, you know, to evaluate the applications within the, you know, within those parameters.
So I will say that CSR does a really good job of putting together our special emphasis panel. They convene many experts, and these are reviewed in different buckets. So there isn't crosstalk between the FOAs when they're under review. We will review all of the lead ID FOAs together as a separate bucket, and most times even on a, you know, at a separate part of the day. And then the lead optimization FOAs are reviewed. And of course, you know, we can't go into great detail about what happens at review, but guidelines are given before each review section begins, and the different criteria are met within review.
Okay, thank you. So now we have a couple of questions that are about, I guess, about investigators. So one is on one of the U01 slides, you suggested that applicants find experienced collaborators. First, is it encouraged for new investigators to apply for U awards as opposed to R21s, or to co-PI a U award with an experienced PI?
Okay, so I would say that we don't have a preference of whether a new or early stage or experienced investigator like comes in with a multi-PI award or comes in as the principal investigator. We are always ecstatic when we get early stage investigators and new investigators. And we encourage all investigators, of course, to apply. And the review is largely dictated by the science that comes in. We are suggesting you find experienced collaborators because that's one of the things we think helps PIs move from academics to industry. Sometimes that's a hard bridge to cross, and experienced collaborators who have done this before, regulatory specialists, industry partners, these are all components of an optimization U01 application that would strengthen the case that you can actually meet your end goals.
Yeah, I mean, I guess the best advice I can give is let's have a one-on-one, because it's going to depend exactly where you are when you say inexperienced. You know, in some cases, you know, we are very interested, as she said, in new investigators. We even have a specific R21 program for that. But you know, yeah, it's going to depend on lots of different things, and it's probably best for a one-on-one conversation. Yeah.
Okay. So the next question is, for early investigators, such as postdocs, are there transition awards or mechanisms such as a K or T grant? Not all institutions allow postdocs to apply for R grants.
Right. Unfortunately, the answer is no right now. There are some restrictions on what we can and do with this special congressional appropriation funds. I can say that I wish we did, and I'll keep working on it. but right now the real answer is no. But like you said, and it depends on the different. Some institutes actually allow postdocs to even apply for it, but very few. But what I would suggest is to go to your IC website and look at their Ks and look at their Fs and look at their other programs like that. And if you decide to, I don't know, maybe you could apply to that and get a good score, and maybe you know, we could support it somehow. But the answer to the question is unfortunately we don't have those.
Okay. So we're nearing the end of the questions. We have 3 left. If you have been waiting to see if someone else is going to be asking your question, it's unlikely. So go ahead and type it in. So the next question is, for animal models, does that include naturally occurring diseases in animals that have human counterparts?
You got me on that one. Not sure I understand the question, but.
Okay, maybe it'll be that if you have those questions, you can email and we can set a time to follow up and talk about it. Okay, one question is whether we have the links to the R01 and R21s mentioned. So I will tell you the R01s that you might be talking about are the IC specific led FOAs, and those links are going to be in the slide deck that we're going to share with everyone afterwards. For the R21s, you can definitely get those at, you know, from, if you go to the CounterACT website and you click for our funding opportunities, our active funding opportunities, there will be links for all of the R21s and the U01s and the U54 active funding opportunities that we have. There was a question about for one of the IC specific FOAs, what the term limited competition means. The best thing to do is to contact the program officer for that was for the IC, that was for the NIAMS related funding opportunity. And so the best thing to do is to contact that program officer directly, and they'll explain it to you. With that, that brings us to the end of our questions and also the end of our time. Okay, one last question has just snuck in, and this is the end of our time, so this will be the last question. This person says that they have an SBIR phase 1 grant to screen opioid receptor modulators. Yeah. Can these be? Can they use the hits of the phase 1 to, as you know, results to apply for one of the U01s?
Well, yeah, I'll be happy to answer this one. And I'll just start by saying we are very, very interested in grants for the opioids. We have a notice or some sort of announcement, and you can find the link on our website where we explain exactly what we mean by opioid threats and how to develop medical countermeasures for them. And I encourage you to read that because we are talking about something very different. We have a whole institute called the National Institute on Drug Abuse that covers individual opioid overdose. We are talking about some special situations. If you want to Google the Russian theater event that happened many years ago now with opioids, it's where not 1 or 2 people have overdosed, but 100 or 200. And in fact, it was 800 people at that time. And that's a good sort of way to look at how we use it. We're talking about the release of opioid to many people at once by a variety of different kinds of ways of the routes that they use. And we're talking about not treating one person, but treating 100 people at once. So we're talking about medical countermeasures that can be used in these mass casualty situations. We're talking about medical countermeasures that are going to be effective to the more potent opioids beyond fentanyl. But again, I think this a good resource is to just look at our notice online. But I'd be happy to talk to you and our representative from the National Institute on Drug Abuse if you'd like to talk more about that.
Okay, so that brings us to the end of the webinar. I'd like to thank you all for joining and for your attention. And I will follow up with all registrants to send them a copy of the slides. Thank you again. And now this webinar is over.